Many of the most devastating diseases that plague humanity are the result of genetic mutations. There are two types of autosomal or non-sex-linked mutations. Substitution mutations occur when a single nitrogen base is altered, changing only the amino acid in which it occurred, whereas frameshift mutations occur when a nitrogen base is added or deleted, affecting all amino acids after it. These are far more severe. Any mutation could be silent, meaning that it has no effect on the phenotype, could alter it in a harmless way, such as heterochromia iridium or red hair, or may alter the phenotype in a way that harms the individual, such as in hemophilia. Mutations can also be beneficial, meaning that they improve an organism's chance of survival. An example of this is lactose tolerancy in humans. These mutations drive evolution. There are also different types of sex-linked disorders. These are caused by non-disjunction, in which chromosomes fail to separate properly during anaphase of meiosis, resulting in gametes with the wrong number of chromosomes. The enormity of such a mutation is clear when compared to the chromosomes present in normal cells, with N representing the number of chromosomes normally possessed in an organism's gametes, such as a haploid cell having N chromosomes and a diploid cell having 2 N chromosomes. It is seen that non-disjunction can take away a chromosome in monosomy, add a chromosome in trisomy, or even triple the amount needed in a triploid cell. An example of a condition caused by non-disjunction is Turner syndrome, in which a person has only one X chromosome instead of the normal 2 X's or an X and a Y. Sex-linked disorders such as Turner's are often followed via pedigrees, diagrams used to track a trait's presence in the family. Sex-linked traits often show many female carriers passing it to sons, but pedigrees can also track autosomal diseases such as cystic fibrosis, and in both cases, dominant traits will be seen in all generations. However, there are some traits that are reliant not only on one's genes, but their environment. For example, the color of hydrangea flowers is determined by the acidity of the soil they grow in. An example of this same phenomenon in humans is lung cancer, a mutation that can be brought on by the toxic chemicals inhaled when smoking. It can be difficult to determine when traits are inherited or a result of one's environment. Twin studies are key to answering such questions, as two individuals with identical genetic information may have different characteristics depending on the environment they live in.